Carsten Timpe from Hoffmann-La Roche, early phase, solid dosage form development at Hoffmann-La Roche, with heading late phase activities, and we'll talk about nanotubes for oral dosage forms.
Yeah, thank you very much for the invitation and Bernd for the introduction. So I gave this presentation at the Science and Innovation meeting we have on a monthly basis, and one of the colleagues was asking, but we do not have this in our excipient stock, so do not wonder. I mean, this is really nothing much we have in development now. So it's an approach which might be attractive in the next couple of years, but nowadays we are not using this. Okay, so a brief overview about my talk. So I would like to draw your attention about the types of nanotubes which are currently described in literature. A little bit talk more in detail about the hallocyte nanotubes and multiwalled carbon nanotubes, and briefly give you a view on the biocompatibility, and then just the main focus of my talk to show you a few examples which might be attractive also for drug development. So this is a table which shows a comparison between nanotubes, hallocyte nanotubes, which are clay nanotubes, and carbon nanotubes. So they are small particles, so 50 nanometer to 1 micrometer, the clay nanotubes, whereas the carbon nanotubes are have a smaller diameter. Whereas the hallocyte nanotubes are viewed as biocompatible, typically the regular non-oxidized carbon nanotubes are viewed as toxic. They have a very large aspect ratio and viewed as toxic. As they have, for instance, like asbestos, they have this fiber problem. So the price is extremely different. So these clay nanotubes are very cheap. They are generated in mines, whereas carbon nanotubes are much more expensive. So the production per year is 50,000 tons on the clay nanotubes and just a few grams on the carbon nanotubes. They have a very special property, the conductivity. So you can coat these clay nanotubes, whereas the carbon nanotubes, they have an sp2 hybridization, so they have a high conductivity, even higher than copper, for instance, and also both are characterized by a high specific surface area. So this more in detail shows you the clay nanotubes, the hallocyte nanotubes. They are named after a Belgian geologist who lived in the 19th century, and there are deposits of this mineral in China, France, Belgium, and New Zealand, and there's a lot of variability because it's a natural source, and currently there are a lot of technical applications described in literature, anti-corrosive, paramagnetic coatings, but also quite interestingly biomimetic nanoreactors. So it's an aluminum silicate. You see here the structure. They can exist as single wall or multiple wall layers. There's a water layer in between those layers, and more or less it's chemically similar to kaolin or identical. The special property is that the surface is negatively charged, the outer surface, whereas the inner surface is positively charged. So, for instance, you can load the core with some negatively charged enzymes like urease, which is then used as a biomimetic nanoreactor. The multi-walled carbon nanotubes, I'm focusing here on the oxidized carbon nanotubes, and nicely you have seen this example here from the regulatory representative from China, this gadolinium compound. So it's more or less related to this fullerenes, which had huge interest 30 years ago, 20 years ago circa. But typically they are toxic for medical applications, but if they are oxidized under harsh conditions with sulfuric acid and nitric acid or with ozonolysis, then you can have biocompatible carbon nanotubes. And here the multi-walled carbon nanotubes are of interest. A nice paper has just recently been published in April this year, a nice review. And with this functionalization by hydroxy and carboxylic group, you can bind here also drugs. So it's now also interesting for pharmaceutical applications, and different binding mechanisms are described, ionic interactions, H bonding, etc. So regarding the biocompatibility, this is here from a paper published a few years ago by an Italian working group, and they have just checked the biocompatibility of the hallocyte nanotubes with some breast cancer or cervix cancer cells, and they found that they are quite compatible up to a certain concentration, which is roughly in the range of 100 microgram per milliliter. So it's quite compatible and does not have the disadvantage of asbestos, which have longer fibers, so they are much shorter. Now switching to the pharmaceutical applications, so there are a lot of papers in the meantime. Just to pick one example for controlled release, this is a paper which has been written 10 years ago. In this case, oxytetracycline has been bound to the lumen of the nanotubes, and you see here a prolonged release over many hours, so up to 30 hours, constant release out of the lumen of these nanotubes. Similar to these clay nanotubes, this also has been described now for these oxidized multiwalled carbon nanotubes. Carvedilol has been chosen as a model drug, and here the release was over 6 hours, 6 to 7 hours approximately. And as for the clay nanotubes, the drug can be bound then either internally or at the outer surface. You see here an example of the oxidized fullerene structure with some carboxylic or hydroxylic groups. So just to focus here on the diameter, so the oxidized multi-walled carbon nanotubes, they have an inner diameter of around about 6 nanometer and length of 800 nanometer, so they are, they have a very high aspect ratio. So, what makes the multi-walled carbon nanotubes, the oxidized, very interesting is that they can have some conductivity. This is an example of a hydrogel where, under the influence of a certain voltage, the drug release can be promoted, and in this case, ketoprofen was used in a hydrogel, and the drug release could be boosted up to 85% with these nanotubes, in comparison only 47 without. In taste masking, it's interesting. So it's an example where sildenafil was bound to such clay nanotubes-like structure. For the sake of time, I will skip this. So what you can see here, in the buccal mucosa, there was just with these nanotubes, there was just a little release of the drug, of the model drug, and then once it went to the stomach, the release was then as expected. So it's an application that might be useful. Of course, it would compete against existing technologies in pharmaceutical industry. This is just a cartoon showing that you can, of course, chemically derivatize the hydroxyl functions. There are papers dealing extensively with this. And last but not least, very interesting, the nanotubes can play a role in bone tissue engineering. So there are papers published where this was combined with PVA and some cross-linking. So just by adding a little amounts of nanotubes, you can dramatically increase the performance of those products. This is an example where nanotubes were added and the mechanical performance was hugely improved. So the tensile strength and elongation at a break was dramatically increased just by adding 7.5% of the clay nanotubes. This is more or less once we are coming now to the end. So another interesting approach is to load these clay nanotubes with drugs and then to combine it in an electrospinning process with polymers, with PLGA, polylactic glycolic acid, and then you can have such fibers, such fibrous mats that could be incorporated in human tissues, for instance, for wound healing. Now this is my last slide. So this is an overview which shows you the potential application of these nanotubes, so the clay nanotubes or the oxidized carbon nanotubes in pharmaceutical industry. It could serve as a matter of stabilizing pharmaceutical drugs, maybe also proteins. It could be used for pH-dependent drug release, controlled release delivery, what I've shown, delivery of antibiotics, these wound healing opportunities. Taste masking could be interesting for pediatric or geriatric application. Site-specific absorption, so with dermal applications in bones and tissues. And it could help to incorporate a very low dose in a fixed-dose combo product. But again, I have to emphasize these are just things which are plans for the future. The products have not been really approved or cannot be easily used as excipients. And based on the introduction of other new excipients into the market, you can estimate that would take at least 7 to 10 years maybe to see those excipients really being used in industry. And of course, not to forget, it always competes against existing technology. So despite the multiple number of papers, the application still is unfortunately very limited in pharmaceutical industrial practice. Thank you very much.
Okay, thank you very much, Carsten, for this nice presentation. Do we have questions on the floor? Maybe one question from my side. Is there any regulatory status assigned to these, like oral safety or?
To my knowledge, not. I think especially the carbon nanotubes, the non-oxidized, they are of course no-goes. I mean, they are in technical applications worldwide now under investigation because they have this conductivity from golf balls to automobile products. But you have to oxidize them, and I think it's also not, the technologies to produce them are not so much validated or robust. Regarding the clay nanotubes, I see this probably more favorable because we have other silicates and aluminium excipients in the pharmaceutical industry. Probably the hurdles are lower. I'm talking about oral delivery, not other
sure.
applications.
Okay, thank you very much. Yeah, the next talk is